Macroevolutionary dynamics and ichthyological systematics, origin, radiation, and adaptive strategies of global fish populations. The biological success of fishes is defined by their unparalleled taxonomic diversity and ecological plasticity, comprising over 34,000 species that represent more than half of all extant vertebrate life. This diversification is the culmination of over 530 million years of evolutionary experimentation, beginning in the Cambrian explosion and progressing through fundamental genomic and morphological revolutions. Evolutionary origins and the Cambrian vertebrate transition. The emergence of fish-like organisms marks the transition from sedentary or semi-mobile protochordates to active, predatory vertebrates. This transition occurred approximately 530 million years ago, primarily during the Cambrian period. The initial chordate body plan, characterized by a notochord, dorsal nerve cord, and pharyngeal slits, underwent a process of cephalisation and ossification that led to the first craniates. The earliest chordates and craniates. Fossil evidence from the Chengjiang biota, particularly the genera Ichthyological and Mylocunmingia, provides a window into the earliest stages of vertebrate evolution. These organisms exhibited serial gill structures and a rudimentary brain case, yet they lacked the complex skeletal systems found in later lineages. Ichthyological is especially significant for possessing a defined skull and evidence of a vertebral column, positioning it as the most primitive known vertebrate. These early forms retained the notochord into maturity, a trait that would be modified as the vertebral column took on the primary role of axial support. The ostracoderm radiation and dermal ossification. By the late Cambrian and throughout the Ordovician, the first major diversification of jawless fishes occurred with the appearance of the ostracoderms. These fishes were characterized by extensive dermal armor, thick bony plates and scales that covered the head and trunk. The evolution of dermal bone in ostracoderms was not merely a defensive adaptation against the predatory eurypterids of the time. It served as a critical metabolic reservoir for phosphate, a necessity for high-activity lifestyles in nutrient-variable environments. Ostracoderms, despite their lack of jaws, exhibited complex sensory systems, including lateral eyes, a pineal eye for photoreception, and a single median nostril. Their circulatory systems were closed, featuring a two-chambered heart composed of an atrium and a ventricle, which established the basic vertebrate cardiovascular blueprint. The retention of the notochord alongside a partially enclosed vertebral column suggests a transitional state of axial development. The following table shows major early lineage, period of origin, primary evolutionary innovation and extinction driver of Anatha, ostracoderms, acanthodii, and placoderms, the Gnathostome revolution and the Devonian expansion. The Devonian period, often referred to as the Age of Fishes, witnessed an explosive increase in fish variety, driven largely by the emergence of jaws. The evolution of the jaw is perhaps the most significant functional innovation in vertebrate history, transforming fishes from passive filter feeders or substrate sifters into active, high-level predators. Mechanics of jaw evolution. The neoclassical hypothesis for the origin of jaws posits that they evolved from the first and second anterior branchial arches, gill arches. Initially, these arches were likely modified to enhance ventilation, allowing for a more powerful pumping mechanism to move water across the gills. This increased respiratory capacity was later co-opted for the grasping of prey, a clear example of exaptation in evolutionary biology. As jaws became associated with dentition, the trophic niches available to vertebrates expanded exponentially. Dominance of placoderms and acanthodians. Placoderms represented the first dominant group of jawed vertebrates, characterized by a unique joint between the armored head shield and the trunk, which allowed for a wide gape and immense biting forces. Dunkleosteus, reaching lengths of 7 meters, exemplifies the apex predator role these fishes occupied in Devonian marine ecosystems. Concurrently, the acanthodians, or spiny sharks, appeared. These fishes possessed jaws and teeth in some lineages, but were distinguished by multiple paired fins, each supported by a robust bony spine. They are often considered a sister group to the modern bony fishes, Osteichthyes, or a stem lineage that eventually led to the Actinopterygii. Their streamlined bodies and multiple fins suggest they were among the first fishes to achieve high levels of maneuverability and speed in the water column. Phylogeny and divergence of modern ichthyofauna. The extinction of placoderms and the decline of ostracoderms by the end of the Devonian paved the way for the two lineages that dominate the modern world: the chondrichthyes, cartilaginous fishes, and the osteichthyes, bony fishes. Chondrichthyes. Cartilaginous specialization. The chondrichthyes, encompassing sharks, skates, rays, elasmobranchii, and chimeras, holocephali, represent a highly successful evolutionary pathway focused on weight reduction and sensory acuity. While once erroneously thought to be primitive because of their lack of bone, it is now understood that their cartilaginous skeleton is a derived feature, a secondary loss of ossification from bony ancestors. Their skeletal structure consists of prismatic calcified cartilage, which provides durability without the mass of a purely bony skeleton. This adaptation, combined with a heterocercal tail and stiff, wing-like pectoral fins, allows cartilaginous fishes to generate dynamic lift. However, the lack of a swim bladder requires them to maintain forward motion or utilize high concentrations of lipids in the liver to achieve near-neutral buoyancy. Osteichthyes, the bony fish matrix. The osteichthyes are characterized by a heavily ossified endoskeleton and the presence of an operculum that covers the gill openings. This group diverged into two fundamental clades. Actinopterygii, ray-finned fishes. The fins are supported by flexible lepidotrichia, bony rays. This group underwent several radiations, culminating in the teleostae, which comprise approximately 95% of all extant fish species. Sarcopterygii, lobe-finned fishes. These fishes possess fleshy, lobed fins joined to the body by a single bone, homologous to the humerus and femur of tetrapods. This group includes the lungfishes, dipnoi, and the coelacanths, Actinistia. The following table shows class, skeletal composition, buoyancy mechanism and primary habitat of chondrichthyes, Actinopterygii, and Sarcopterygii. Genomic foundations of teleost diversity. The immense diversity of teleost fishes, exceeding 30,000 species, is primarily attributed to the teleost-specific genome duplication, TGD. This whole genome duplication event occurred in the stem lineage of teleosts approximately 320 to 400 million years ago, following their divergence from non-teleost ray-finned fishes such as gars and sturgeons. Neo-functionalization and the root effect. The TGD provided duplicated sets of genes, paralogs, that could evolve new functions through neo-functionalization or subfunctionalization. A quintessential example is the evolution of hemoglobin, Hb, isoforms. Teleosts possess multiple alpha and beta globin genes that allow them to produce functionally distinct hemoglobins adapted to different ontogenetic stages and environmental conditions. The root effect is a specialized property of teleost hemoglobin where binding capacity is extremely reduced at low pH, even in the presence of high oxygen tension. This is a key evolutionary innovation derived from TGD-related gene diversification. The root effect is essential for buoyancy, secreting oxygen, O2, into the swim bladder against high-pressure gradients, retinal oxygenation, delivering O2 to the avascular retina, which has high metabolic demands, thereby facilitating superior vision in deep or murky waters. The TGD also influenced the diversity of the mobilome, transposable elements, although current research suggests that while TEs are highly diverse in teleosts, their expansion was more lineage-specific rather than a single burst following the duplication event. Global distribution and zoographical realms. The distribution of fish groups is not random but is the result of intricate interactions between plate tectonics, climatic shifts, and historical dispersal events. Tectonism and the Indo-Pacific complexity. The Indo-Australian archipelago serves as a prime example of how geological evolution shapes biodiversity. Large islands such as Sulawesi, New Guinea, and Borneo are biological and geological composites. Tectonic shifts during the late Jurassic, 160 MYA, caused New Guinea to rift from Sumatra, and later, bits of Borneo and Sulawesi were carried by tectonic plates, only to converge millions of years later. This historical rifting created vicariance events that isolated freshwater fish populations. The closing of the ancient Tessis Sea resulted in suture zones like Hamilton's Line, which marks the biological transition between distinct faunal realms better than the traditional Wallace's Line. In many cases, the distribution of freshwater fishes is more closely aligned with the boundaries of major ocean basins, Indian, Pacific, Atlantic, than with present-day continental boundaries, as the oceans acted as the historical conduits for transcontinental biotic tracts. Case study: Cenozoic evolution of Nemachelidae. The stone loach family, Nemachelidae, provides a detailed model for the evolution of Eurasian freshwater biodiversity. This family originated in Indochina around 48 million years ago. Their distribution expanded across Eurasia through tectonic connections and favorable climatic conditions. A major aridification event in Central Asia between 34 and 23 MYA acted as a geographic divide, splitting ancestral groups into separate clades that evolved in isolation. Furthermore, the West Burma terrain acted as a ferryboat, transporting fish from Southeast Asia to Northeast India around 30 MYA, which then allowed for colonization into the Near East and Europe. This demonstrates that the current distribution of freshwater fishes is a map of the movement of the Earth's crustal plates. River erosion as a driver of speciation. While tectonically active regions like the Himalayas are known hotspots for biodiversity, research indicates that tectonically quiet regions can also drive evolution through river erosion. In the Appalachian Mountains, as rivers erode through different rock types, they alter the landscape and push fish populations into different tributaries. This process isolated populations of the greenfin darter, leading to significant genetic divergence even where outward morphology remained similar. This highlights that landscape evolution, even in the absence of active plate movement, is a primary driver of allopatric speciation. Evolutionary strategies in morphology and locomotion. The aquatic environment imposes severe physical constraints on movement, leading to a variety of morphological strategies for navigation and stability. The evolution of the swim bladder. The swim bladder is an unpaired dorsal outgrowth of the posterior foregut, evolutionarily homologous to the lungs of tetrapods and lungfish. Early bony fishes likely possessed a ventral lung to gulp air in oxygen-poor shallow tropical waters. As lineages moved into deeper or more stable environments, this lung was adapted into a dorsal buoyancy organ, the swim bladder. Physostomus fishes retain a connection, the pneumatic duct, between the gut and the swim bladder, allowing them to fill the bladder by gulping air at the surface. Physiclistus fishes have lost this duct. The bladder is inflated and deflated via the gas gland and the rete mirabile, which utilizes the root effect to move gases from the blood into the bladder. The transition to a dorsal swim bladder provided neutral buoyancy, which allowed teleosts to repurpose their fins. Unlike sharks, which must use their fins to generate lift, teleosts can remain stationary or swim slowly, using their fins for precision maneuvers, such as turning on the spot or swimming backwards. Fin morphology and design. Ray-finned fishes possess 4 key design features in their fins that are critical for navigating complex habitats. Flexibility. Fins can bend to resist or utilize fluid forces. Collapsibility. Fins can be folded against the body to minimize drag during high-speed swimming. Bony support. Fin rays, lepidotrichia, are supported by jointed segments. Bilaminar structure. Each fin ray consists of 2 halves, hemitrichia. Differential muscle activity at the base of these halves creates curvature, allowing the fish to actively control the fin's surface area and shape. Trophic strategies. Biomechanics of prey capture. Feeding is a complex behavior where morphology and hydrodynamics must be integrated to overcome the escape responses of prey. Suction feeding versus ram feeding. Suction feeding is the most common mode of prey capture in fishes. It involves the rapid expansion of the buccal cavity, which creates a negative pressure gradient that draws water and prey into the mouth. This expansive phase is achieved through a coordinated movement of jaw opening, hyoid depression, and cranial rotation. Mechanical advantage. Suction performance is influenced by the gape size and the rate of mouth expansion. A smaller mouth aperture can actually increase the force exerted on the prey by creating a steeper pressure gradient. Ram feeding occurs when a fish swims over a prey item with its mouth open, effectively using its forward momentum to engulf the prey. Tunas and whale sharks are classic examples of ram feeders. Most teleosts use a combination of both, which can be measured using the ram suction index, RSI. The incorporation of jaw protrusion, extending the mouth toward the prey, allows predators to initiate a strike from a greater distance and increases the total force exerted on the prey by up to 35%. The following table shows feeding component, metric of performance and morphological basis with respect to suction pressure, flow velocity, ram speed, and jaw protrusion. Reproductive strategies and life history evolution. Fisheries management relies heavily on understanding how reproductive strategies influence population dynamics and resilience. The Weinmüller-Rose triangular model. Life history theory in fishes is often summarized by a triangular model that defines strategies based on three demographic endpoints: opportunistic, periodic, and equilibrium. Opportunistic strategists. Small-bodied, short-lived species with early maturation and high reproductive effort. They are designed for colonization and are favored in unstable, unpredictable environments. Periodic strategists. Large species with long lifespans, late maturation, and high fecundity, e.g., millions of eggs. They are adapted to large-scale, predictable environmental variation. Equilibrium strategists. Species with moderate to long generation times and high investment per offspring, e.g., large eggs, parental care. They are favored in stable, resource-limited environments where competition is high. The following table shows strategy type, body size, fecundity, parental care and resilience to overfishing for opportunistic, periodic, and equilibrium strategists. Reproductive tactics. Elasmobranch viviparity. Elasmobranchs exhibit highly advanced reproductive modes, ranging from oviparity to placental viviparity. Lesotrophic viviparity. Embryos develop in the uterus but are nourished solely by a yolk sac. Macrotrophic oophagy. Developing embryos consume unfertilized eggs produced by the mother during gestation. This occurs in all lamnoid sharks. Placental viviparity. A yolk sac placenta forms, allowing direct nutrient transfer from the mother to the embryo. This evolved independently 11 to 20 times in elasmobranchs. Lipid histotrophy. The uterine wall secretes uterine milk, histotrophy, that is ingested by the embryos, a strategy common in stingrays. Sensory systems and adaptive communication. Fishes have specialized sensory organs that enable them to navigate and communicate in environments where light is limited. The mechanosensory lateral line system. The lateral line, LL, system consists of neuromasts located on the skin, superficial, or in bony canals. Superficial neuromasts, SNs, act as velocimeters, responding directly to fluid velocity. Canal neuromasts, CNS, act as accelerometers, responding to pressure differentials between canal pores. Cranial lateral line canals are typically associated with a conserved set of bones, but their phenotypes vary significantly based on habitat. For example, widened canals are often found on the functionally ventral side of the head in benthic foragers like the silverjaw minnow, aiding in the detection of prey in the dark. Electroreception and electric organs. Electroreception is a widespread sensory modality, particularly in chondrichthyans and some teleosts. Weakly electric fish, e.g., gymnotiforms and mormyridae, have independently evolved electric organs to communicate and locate objects. This was achieved by duplicating the sodium channel genes in muscle cells. Electric fish then turned off the muscle contraction function and repurposed the ion channels to generate an electric organ discharge, EOD. Objects in the water distort this electric field, and the fish detects these distortions through epidermal electroreceptor organs, a process known as active electrolocation. Physiological adaptation to extreme environments. The colonization of extreme habitats, such as the deep sea or hadal zones, requires biochemical modifications to maintain cellular integrity under crushing pressure. Pressure resistance and piezolites. High hydrostatic pressure, HHP, destabilizes proteins, forcing water molecules into the protein interior and causing denaturation. Deep-sea fishes adapt through the accumulation of piezolites, small organic solutes like trimethylamine N-oxide, TMAO. TMAO stabilizes proteins against pressure by altering the structure of the surrounding water, making it energetically unfavorable for water to enter the protein core. TMAO concentrations in teleost fishes increase linearly with depth, from 40 mole a kilogram at the surface to 261 mole kilogram at 4,850 meters. This correlation suggests a depth limit hypothesis. As TMAO increases to maintain protein stability, it simultaneously raises internal osmolality. At a depth of approximately 8,200 meters, a fish's internal osmolality would become iso-osmotic with seawater, potentially setting the physiological depth limit for vertebrate life. Visual adaptations in the mesopelagic zone. In the twilight zone, 200 to 1,000 meters, light is extremely faint. Deep-sea fishes have evolved eyes with larger lenses and retinas optimized for photon absorption. Bioluminescence is ubiquitous here, used for camouflage, counterillumination, predation, and mate attraction. Counterillumination involves ventral photophores that match the downwelling light's intensity, making the fish nearly invisible to predators looking up from below. Future outlook and recommendations for fisheries management. The evolutionary history and strategies of fishes provide a foundational context for modern fisheries science. The Weinmüller-Rose model, for instance, predicts that equilibrium-type species, like sharks, will be highly sensitive to overharvest due to their low natural recruitment and long generation times. Conversely, periodic-type species, which dominate commercial fisheries, are subject to high interannual recruitment variation driven by large-scale abiotic factors, making them harder to manage using simple density-dependent models. Management strategies must transition toward a total evidence perspective that integrates molecular phylogenetics to resolve taxonomic ambiguities and identify cryptic species. Life history ordination to predict population resilience and set sustainable harvest levels based on species-specific demographic traits. Zoographical mapping to identify critical migratory corridors and historical refugia that are essential for conservation in the face of climate change. Thank you for watching.